I would like to welcome you all to today's webinar on behalf of Interherence. My name is Jaroslav Icha, and I will be the event host. I'm in charge of marketing activities at Interherence. And before I give the word to our two scientific speakers, let me tell you about the schedule of today's event and say also a few words about what we are doing in our company. So we will start with the talk by Senthil Arumugam, followed by Q&A specific to his talk. Then Alex Bisson will take over with his presentation. And after Alex's talk, let's have specific questions to his talk first. And then we can move on to more general discussion about lifestyle imaging, about temperature control in microscopy. But we will finish latest at 5:10. I have also two of my colleagues here, our CEO Pierre and our CTO Daniel. So you can be sure that your questions will be answered. Now, the questions, preferably, please ask them via chat, or you can also raise your hand after the talk, and we will let you unmute yourself and pose the question. So a little bit about us. Interherence is based in Erlangen, Germany. We are a spin-off from the local Max Planck Institute in Erlangen for the science of light. We are in business since the beginning of 2020, and we keep growing, and most of our manpower is currently focused on R&D of the next products. In our team, we have mainly physicists with a lot of experience and knowledge in microscopy. The first product that we launched last year is a temperature control device called VAHEAT. The second product will be launched very soon. And so what is our mission in general? We want to make high sensitivity microscopy accessible to everyone. And we design our systems with ease of use in mind, and also with reproducibility issues in mind. So we want to make your imaging conditions as reproducible as possible. And all our devices contain some element of integrated electronics and optics. Here you can see our nanofabrication expert Atip working in the clean room on the prototypes of our next chip-based devices. But let's quickly mention what we already offer. So that's VAHEAT, our solution to common challenges in temperature control in high-resolution microscopy, especially. Thanks to this functionalized cover slip that you see on the right, that contains both the heating element and the temperature probe, you can control very precisely the temperature of your sample directly in the field of view. This device has already found many applications in lifestyle imaging, and especially the community around Archaea, around imaging extremophilic organisms, is quite fond of this instrument. I have chosen two publications as distinct examples of what VAHEAT can be used for in lifestyle imaging. So the first one is from Wolfgang Zacharia lab, where they used VAHEAT to heat shock and simultaneously perform lifestyle imaging. The second example is from the lab of Guillaume Bafou, where they used VAHEAT for the already mentioned application of imaging extremophilic organisms. But let's move on to our scientific speakers. So the first one will be Senthil Arumugam. He's joining us from Australia, where it is already after midnight. So Senthil is up late, and he also has a newborn child at home. Congratulations. So these are pretty extreme lecturing conditions, I would say. So Senthil originally comes from India, where he studied chemistry and biophysics. And after that, he did his PhD with Petrashvili, focusing on self-organization of proteins involved in bacterial cell division. And that is something he shares with our second speaker, by the way, the focus on bacterial cell division. Senthil did his postdoc at the Curie Institute in Paris, where he already started working on topics that he focuses on nowadays, namely membrane trafficking. He has an independent research group in Australia since 2016, and since 2019, he holds a position as an EMBL Australia group leader at the Monash Biomedicine Discovery Institute. He established the lattice light sheet imaging infrastructure there, and he focuses on fundamental questions in intracellular trafficking and its role in sulfate specification and pattern formation. So Senthil, we look forward to your talk.
Thank you, Jaroslav. I can share my screen. Right. Can you see that all right?
Yes.
Yeah. Thank you very much, Interherence and Jaroslav, for the kind invitation. I'm 21 hours awake, so if I make any mistakes or if it's confusing, ask anytime or send me an email and I'll make sure that I respond to you. Right. So I'm going to talk about a topic that we've been exploring in the lab for the last 4 or so years. Before I proceed, I want to thank the actual people in the lab who did the work. So that those 5 are my lab, and there's a bit of help from image analysis and simulations that comes from Iyer Biswas lab at Purdue University. And we take a lot of help from Ulrike, who's at the University of Bonn, for a specific tracking algorithm that I have found to be the best one that works to date in a very complex data set. I'd also like to thank SciGlass, with whom we've been working some virtual reality based visualization and loading of analyzed data to better infer what's in the data, because these are complex sets of data, as you will see. So I started my group at UNSW Sydney, as Jaroslav mentioned, and I'd like to express my gratitude to Kat, who unfortunately passed away this year. She was a friend and a mentor. She was the one who recruited me to Sydney during these difficult lockdown times. Couldn't meet her, but yeah, it's because of her and her vision that we could get lattice light sheet and everything going on, and specifically these projects that are working today are because of her. Right, so let me get into the science. So that's the standard lore of endosomal network. That's what you see in textbooks. You get endocytosis at the plasma membrane. It can be clathrin dependent, clathrin independent. They form vesicles that fuse on to form subsequent, more mature vesicles. These are defined by the kind of proteins on them, mainly the RAP proteins. So if you have RAP5, they're termed as early endosomes. If you have RAP7, they're termed as late endosomes, and so on. So that's the network of endosomal pathways, right? But the processes that build up this network are highly stochastic. What do I mean by that? I mean that these endosomal network undergo continuous fission and fusion with each other. The vesicles are being moved by motor proteins, kinesins and dynein, move them to and fro within the cell. The membrane specificity of is provided by the presence of phosphoinositides, which they themselves are in low numbers, and therefore the binding affinities are stochastic in nature. And then there are enzymes that bind and convert these phosphoinositides from one form to another, right? And all these things are interlinked somehow, and these give rise to this very robust network of organization as we know it, right? So the question is, since individual processes that build up this network are stochastic intrinsically, but when you look at a certain cargo or receptor signaling, receptor transport, you see very deterministic outcomes, right? So then the question is there a mechanism, or are there multiple mechanisms that make these processes very robust? And so we would, as we do in the lab, we start with imaging the system and to capture processes and hints about what could be happening there with respect to specific molecules or a particular dynamics of the system. So typically, the process of endocytosis has been well studied because it's closer to the plasma membrane, and you could use, for example, total internal reflection fluorescence microscopy, which can give you very good resolution and good signal to noise at the cover slip. And you can image processes like endocytosis or exocytosis very, very clearly. However, when you want to image these processes deep within the cell, you have to be aware that you need good spatial temperature resolution, but you also need very good time resolution because these vesicles are moving at 1 micrometer per second as being pulled by kinesins or dynein. And you want to image the entire volume of the cell because they may be moving in and out of the plane. And then you need to image for long enough time because you could be imaging stochastic processes. So you don't know when they're going to happen and where they're going to happen. So you want to image long and fast, which means photo gentleness becomes a very important aspect of capturing these processes accurately. And so therefore we use lattice light sheet. The advantage is that you get a very thin light sheet that's of the order of the wavelength of light, and your excitation thickness is perfectly matched with the detection depth of focus, right? And that means that you're completely exciting that one plane at a given point of time that you're imaging, and you do not have any out of focus excitation, which results in high photo gentleness, right? And so what you do is then you scan the cell as shown in this original manuscript video. You scan this cell through this thin sheet of light repeatedly. So you get one volume and you get another volume and another volume, and therefore you get a volumetric movie out of it, right? So here's an example. So the first time I encountered this microscope was at Tom Christensen's lab where we did that. And this is at UNSW Sydney after we established that. And so these are two different samples, different proteins, but essentially we could get similar quality and similar operations going on in Sydney and now at Monash, right? I'm not going to talk a lot about the analysis routines that we're developing because of limited time, but in essence, we're continuously working on segmenting, tracking, and post trajectory analysis with the help of companies, Ulrike and some collaborators. So with this kind of computational analysis, you can capture ensemble events and get nice statistics and overall quantitative measurements of individual events very, very accurately. So I'm going to specifically focus on just two questions. One is that of receptor signaling, and that's already published. So I'm going to just go through the story very quickly. And the second one is that of endosomal maturation. This is unpublished data, fresh data that has been going on in the lab through the lockdown years, let's say the last 1.5 years or so. So EGFR is present at the plasma membrane. When it is not stimulated, it goes to the early endosome and back to the through the recycling endosome to the plasma membrane. When you stimulate them with EGF, they oligomerize and they start signaling because of phosphorylation in the tail. This signaling stays on until they get into the perinuclear region where there are phosphatases that switch off these. There are phosphatases which essentially switch off these receptors, right? And so one could imagine that the plasma membrane is on, and when it reaches the perinuclear region, it's off. And then the time that it takes for a population of receptors to travel from the plasma membrane to the perinuclear region sort of determines the time for which these ensemble receptors will be switched on. And that means it's modulated by a lot of adapters, proteins, and motility that will define as a population how many receptors reach the perinuclear region as a function of time, right? And so that's where imaging things at 37 degrees comes into play, because a lot of papers in biology describe getting the cells to 4 degrees, adding the ligands, and then bringing them up to 37 degrees. What we did here was we injected the ligand directly while the imaging was performed. So you could see in the transferrin channel, you could see the transferrin ligand come up, and it gets internalized and it starts localizing into various endosomal populations, right? And this is important because a lot of things are taking place at 37 degrees that otherwise do not take place at 4 degrees. In this example, we see that the APPL1, one of the adapter proteins for EGF, immediately co-localizes with the EGF receptors, and then they co-traffic in a way that the entire population of the EGF bearing endosomes reach the center of the cell within 10 minutes. And that's because you have calcium currents. When EGF binds, you can see the signal go up, you get calcium currents, and those calcium currents are important because that causes this APPL1 to dissolve from the pre-existing endosomes that are present and go and bind to newly phosphorylated EGF receptors. And then they engage dynein and bring them to the center of the cell, right? So this is an example where imaging at 37 degrees is absolutely essential so that you have the physiological calcium currents and therefore all this subsequent adapted protein action involved. So here's just multiple Z sections through the cell, and you can see that when EGF is added, the center of the cell, which has some endosomal signal, completely disappears and goes to the outer plasma membrane and comes back into the perinuclear region. So essentially, this is the story. You have pre-existing APPL1 endosomes. When you have EGF added, you get calcium current spikes, which results in desorption of APPL1, and they have a phosphotyrosine binding domain, so they go and bind the EGF receptor specifically, engage dynein, and bring them as a cohort movement to the center of the cell, right? So that's an example of timekeeping through receptors. The second example I want to show is the endosome themselves mature with time. So if you start with, let's say, a very early endosome, they're defined as late endosomes after some time, right? So what sets the rate of these endosomal maturations is what we asked here. So the molecules that we're concerned with are the very early endosomes and early endosomes that are marked by APPL1, EEA1, and RAP5. This is not a novel observation by any means. This has been reported in 2009 by Pietro Di Camilli's lab. The drawback is that there's only a handful of events, right? One does not know how many of these events take place in the cell, and are there any other sophisticated behaviors, because these are probably a few events that slow imaging in one single plane could capture, right? And so with lattice light sheet, we could do a whole cell measurement and quantify them very nicely. So before I get into the movies, I want to just talk about the main molecular players. One of them is EEA1. This is the main central player of the story. It's a 200 nanometer long molecule, and it's got C-terminal and N-terminal. Both the terminals bind to RAP5, but C-terminal also binds to PI3P, one of the phosphonestides in the more later early endosomes. The APPL1 has a pH bar domain, so it binds to PI3,4P2, but also binds to RAP5. And RAP5 again binds to both EEA1 and APPL1, right? And so, as I said, EEA1 is the centerpiece of the story, and I'm going to talk about three behaviors that we observed for this molecule. It's collisions, clustering, and cycles. So here's a movie of APPL1 and EEA1. This is now at whole cell level. It's very difficult for me to point out the very specific events in this huge amount of data that we have, but I'm still looking for ways to present and communicate in manuscripts, etc., very, very precisely. So if anybody has got any fantastic ideas, come back to us. We are thinking about using VR for these purposes, right? But anyway, the main observation is that as you see APPL1 endosomes go, stochastically towards the center of the cell, once in a while they collide with EEA1 molecule, and we observe that every time there's a collision, there's most likely a convergent event, which means the APPL1 signal on the endosomes is being replaced with EEA1 signal. So we quantified that, and you could see that the collision to conversion rates are higher and direct conversion rates are lower. The second experiment we did was because these collisions are a result of endosomes moving on microtubules mediated by motor proteins, you could inhibit these motor proteins using C-librivin. Dynein is inhibited by C-librivin, or you could mildly depolymerize microtubules by using nocatazole. In both cases, what you observe is that the number of conversions per unit time per cell goes down, right? And in this movie, you could see wherever the brackets come up, we picked up EEA1 endosomes and APPL1 endosomes sort of meeting with each other and then changing colors as a function of time. And so what we think is happening, remember that I told you that EEA1 is a 200 nanometer long molecule with Rab5 binding sites at both ends. So what we think is happening is when these are colliding, something interesting is taking place there. Right? So here's a zoomed up, a slightly higher resolution movie of an APPL1 endosome that's going to travel and collide with this EEA1 endosome and pay attention to the EEA1 signal on this APPL1 endosomes, right? So I'm going to start the movie. So this endosome is going to go collide with that one. And as soon as it collides, you can see that it's getting some EEA1 signal, and then it results in fusing with the subsequent EEA1 endosome. If you look at the intensity traces, that's the EEA1 signal on the original APPL1 endosome. There's not much. It tries to, there's a little bit of fluctuations, but it really does not get converted. But as soon as there is a collision, there's a spike, and then it starts converting, resulting in complete fusion, right? So there is something interesting happening in the collision. We think that it's physical transfer of some molecules. And the reason we think that there is a physical transfer is because it's got both N-terminal and C-terminal that can bind. So it's essentially doing some sort of a seeding event from one endosome to the other, right? The second thing is clustering. So we fixed these cells and did a storm analysis, 3D storm. And we find that these EEA1 are not uniformly distributed, but they're always clustered. And that makes sense for collision-mediated transfer, because if you're clustered, you probably transfer a quanta of molecules, not just one or few molecules. We still are trying to figure out the physics of clustering for EEA1 molecules. It could be phosphoinositide mediated, or it could be mediated by the fact that these are 200 nanometer long molecules. There could be some nematic ordering. We don't know. We're exploring that, right? But what's more interesting is that if you look at the distribution of APPL1 and EEA1, they're counter-clustered, which means that wherever there is an APPL1 signal, there is no EEA1 signal. And this sort of suggests a negative feedback in the system. And that's not just in space. If you see in time as well, you get this huge EEA1 fluctuations, which is very strange for us. But then whenever the APPL1 signal starts decreasing, you start getting this EEA1 increase. So they're not just counter-clustered in space, but counter-clustered in sort of time as well. So there should be some negative feedback between EEA1 and APPL1. And so then we started looking more carefully into these two domains in the EEA1, because it's N-terminal and C-terminal, and we think both of them can bind to the endosome. So we wanted to map out whether it's N-terminal bound or C-terminal bound as the endosome matures with time, right? And so the strategy for that was we have an N-terminally tagged EEA1 GFP and an RFP tagged Rab5. If it's C-terminally bound, the two fluorophores are about 200 nanometer apart, right? If it's N-terminally bound and EEA1 N-terminal interacts with Rab5, which means they should be within the FRET distance, and they should the donor should show decreased lifetime, right? And so what we see that the newer endosomes have lower lifetime than the later endosomes, which means that we have, this is live film. And so you can see these new endosomes that are formed that have lower lifetime, and as they mature, they get longer lifetime, right? So what this means is that you have N-terminal binding first to Rab5, and then the C-terminal binds to Rab5 and PI3P. So which means there's some sort of cycling. I shouldn't call it cycling because it's not the same molecule. It's probably a different population of molecules. But there is, as a population, there is cycling of N-terminal bound and then C-terminally bound EEA1, right? So what we think is happening is at first there's N-terminal binding, and there's some sort of negative feedback to make APPL1 dissolve from the endosomes. At the same time, there's a positive feedback to recruit more EEA1 onto the endosome. Sorry, these blue dots here should be yellow. So the more specific model in terms of biochemical players is that you have Pi3-4P2 containing endosomes that have APPL1 bound on them. When there is a collision, you plant some N-terminal molecule. These N-terminal molecule are bound to RAP5, so they kind of sequester off RAP5, which weakens the APPL1 binding to these endosomes and leaves off. And that gives rise to free Pi3-4P2 that's converted into Pi3P by INPP4A, one of the dephosphorylating enzyme. And then increased Pi3P results in now C-terminal of EEA1 binding onto them, right? So this initial seeding instigates this conversion, but as a result of conversion, now you have the C-terminal EEA1 binding onto it, right? So we could sort of plug in this model in a simulation, and this is an experimentally observed curve, and we can see that with the clustering and with all the effects that are described here, we could get very similar phenomenology happening in the simulations. And we could also simulate the process of collision that results in instigation of this conversion. And when we do that, you always see that there is a jump in EA1 signal, which you also recapitulate here. The result of that, if you look at that, is the average time it takes for a population of endosome to convert is decreased, right? Or you could also call it the mean first passage time also decreases when you have collisions compared to without collisions. So what our model really is that, if you consider this as 0 and this is 1 sort of identity of the endosome, what the model really is that the 0th endosome that already has C-terminally bound acts as a parent endosome for seeding some N-terminal EEA1 onto the newly incoming endosome. And that results in the sort of feedback loop that I described here, and that we've simulated, and sort of we see that it is able to recapitulate the observed behaviors. And then this can now act as a feed forward for another incoming endosome, right? So this way it keeps on cycling back and forth to newer incoming endosomes, making sure that the incoming endosome receives enough number of EEA1 molecules to trigger the reaction that is needed to keep the conversion going on, and therefore making the distribution shorter in time. What happens to these endosomes? These endosomes then fuse with each other, undergo an entropic collapse, as has been described by Marino Zerial's group, and then it fuses and results in further later endosomal population, right? So that's an ongoing story. It's not complete by any means, but it's fresh, unpublished data. It's exciting for us. The reason why we are interested in VAHEAT is because one part of the lab also works on transcription factors and phase separation. So this is a collaboration with Roger Pocock's lab, where you're looking at mediator complexes in the germline of C. elegans, and you get this nice punctate. And the question is every foci an LLPS, or liquid-liquid phase transition, or not, right? And one of the more stringent experiments in this aspect is to look at temperature. This is a nice critical review of the experiments that are used to prove or disprove liquid-liquid phase transition, and we think that temperature is one of the better ways to show that it's phase transition or not. So the VAHEAT system is really useful, easy to use across many microscopes. In this aspect, we use it for C. elegans, zebrafish, single cells. This is an example of optodroplets. You can do using optogenetics, or you can do a temperature dependent thing, and VAHEAT is really useful. And I would vouch that it's really easy to use. I've done some temperature dependent studies back in 2015 with some homemade system, and I know how difficult it is, and VAHEAT is really extremely easy to use, right? With that, I'd like to finish here and just put on this gallery for everyone to enjoy, and take this opportunity to advertise for a postdoc position between me and Alex DeMarco with some FIPSIM focused on organelles and cell migration. And thank you, Interherence, for the kind invitation.
Thank you, Sanhil, for your nice talk and for the kind words about VAHEAT, and also for sharing some unpublished data. So far, I have not seen any question in the chat. I would have some, so maybe I can ask first. So since you had the phosphoinositids in your model, did you also try imaging with the fluorescent probes for different phosphoinositids in your system? Is it feasible? Is it technically feasible?
Yeah, so there are various probes, and those are essentially those are not probes. They are just the same domains as, for example, APPL1's pH bar domain, right? So it reports on it at the same time. It also influences the system. So one has to be careful about overexpressing and changing the dynamics of the system. But yes, we have tried with mild overexpression, and we see similar behaviors. In our opinion, I didn't talk about a lot of control experiments. In our we have INPP4A knockout cells that are, that wouldn't carry on the phosphoinositide conversions. But one challenging aspect of studying such dynamical systems is that there's steady state, and then there is equilibrium of after you've perturbed the system, right? What you mostly observe when you've knocked out a protein or overexpress something is the final equilibrium that the system has already gone away from its natural state, right? And so in some sense, it doesn't tell you a lot. It just tells you the aftermath of whatever perturbation you're doing, right? So
we would have time for some questions. So there is one question in the chat. How long can you track a molecule or an endosome? For how long?
Okay, a molecule, we cannot track a molecule because we don't have the signal to noise. It's probably better done with EMCCD cameras or something. But generally, we can track the population of all the endosomal markers for about an hour easily with very little photo bleaching.
Okay, maybe I'll allow myself for one more question. So can you tell us how you mount C. elegans for VAHEAT? Because I think you are one of the few labs using VAHEAT for C. elegans imaging.
Yeah, so we mount it using agarose pads. We typically use a silicone oil, slightly longer distance objective, so it will get better resolution. It's the same as mounting on any other slide for C. elegans imaging. It's because the VAHEAT system is like any other slide except for the electronics below it to heat up, right? So it's no different than anything else. Yeah.
So let's take one last question from chat from Philip Tripal. He asks, do you expect similar modulation of the lysosomal motion? And maybe, Philip, can you specify your question?
Yes, thank you. Do you hear me?
Yeah, I can hear you, Philip.
Okay, thank you for your nice talk. And I'm really interested in the movement of the lysosome in neurons, because they recently found out that the RNA body is moving bound on the lysosome, and this might be really important for the neuronal activity. And do you also expect that the modulation of the lysosome is similar to the endosome, that you also have proteins which can flip-flop, or is this, or you expect only that the lipids in the membrane of these compartments can change the motility?
Right. So the first part of the question, do you have similar flip-flop molecules on the lysosomes? It's not known. I mean, some amount of EA1 is present, but by that time it's already done its cycling. The lysosomes are mostly defined by RAP7 and LAMP1. There could be other molecules. I would definitely say that we don't understand everything about lysosomes and stuff. So I guess there is things to discover. Second thing about imaging neurons. Neurons, you could probably get away with imaging on simpler systems like wide field because you get these long processes and the cell body itself is small, so you don't need a light sheet for that essentially. Sorry, there was another question.
Now the lipids in the endosomal membrane, can they affect speed?
Right. Yes. Yes. So if you just use lysotracker or RAP7, so essentially that movie I show this one, right? This one, the magenta is RAP7. That's lysosome. So you see the movement is very similar. It's stochastic. It moves in little bursts and then wiggles for a minute and then bursts again and so on, right? So the movement seems very similar, and it's mediated by both dynein and kinesin. So a lot of aspects of directionality. When does it choose to move in one direction versus the other is not known, right? Lipid-wise, you still have the phosphoinositide conversions going on. The enzymes that are associated are P10 and PA3K kinase, INPP4B, etc., which also generate PI3P, but from a different pathway, right? So that whole meshwork is reasonably well understood, but there's still a lot to discover. The papers you should read for them are from the labs of Volker Hauke and Christina Mitchell. I think we don't do a lot of lysosome work, so I'm not qualified to best answer your question there.
Do you know the work by Lippincott-Schwarz?
Yeah, I know. Yeah.
When they show the RNA body bound to the lysosome moving on the tubulin network.
Yeah.
This is why I was starting to dig in this, because we recently found out that the lipid membrane, like if there's more ceramide, the people have depression, and if they have more sphingomyelin, the people are healthy. So this might be due to the traveling of the lysosome. And I think that people could not find it out because of problems with temporal stability.
Right. I mean, you're going from molecules to behavior. That's a huge scale of things, right? So it has to be careful there. Yep. Yeah, but thank you for your point. Yep.
But I think for all the analysis, when you track organelles, the temperature has to be stable. Otherwise, you have these big differences like a standard deviation, and you will never find out small differences.
Yep.
Okay, but you think if I use neurons, I don't need light sheet techniques. I can do it just in a turf system or a wide field.
I mean, a good wide field system does most of the jobs. But yeah,
all right.
Look, happy to chat over offline. Just send me an email or something. I think we're waiting for Alex's talk. Right?
Yeah, thanks.
Yeah,
exactly. Thanks, Sandhill. But let's move on to the second talk now, which is given by Alex Bisson. Alex originally comes from Brazil, where he studied biochemistry and molecular biology at the University of Sao Paulo. He moved for his postdoc to US and worked with Ethan Garner at Harvard on bacterial cell division. Since 2019, he runs his own laboratory at Brandeis University. He is focusing on cellular organization, cell shape, and cell divisions across the diversity of Archaeal phylogenetic tree. Alex was using VAHEAT this year during his practical at the famous Woods Hole physiology course. So that's why we decided to have him today as the speaker. In general, his lab is very active also in tool development because there are no existing methods for what they are trying to capture live in the microscope. And I bet some of these gadgets will be featured in Alex's talk. So please, Alex, you can go ahead and start your presentation.
Okay, Thank you, Jaroslav. Can you guys hear me? Great. And can you see my presentation now? Okay. Okay. So thank you so much for the invitation. As introduced by Jaroslav, I'm Alex. I'm going to be talking a little bit about the interplay between the molecular level and cellular behavior. So I believe that, this was, briefly mentioned and writing the end of the questions. And of course, it's a huge, challenge to connect what is happening in the nanometer scales, and then in the micro and milli and second lifetime of the cells, and connect to the long-term evolution, how cells are selected. So today I'm going to talk a little bit, it's a brief talk, it's not super technical, but it's around the techniques that we are developing. And then I'm happy to answer any questions about specifics of the techniques. So to just have a grasp, very, very general interest of the lab is that we are interested in cellular and organization and behavior of the cells. What does that mean is that we have a number of different cells that we are interested, and then we start studying how the physical and the chemical world regulate their behavior or their response. And by evolutionary mechanisms, for example, natural selection, they will propagate and self-organize in a certain function. If you think about the human body, we have a very well-studied different systems, different tissues, different cell types. However, this is much less clear for free-living unicellular organisms. And one consequence of this mechanism of selecting and specializing function is that we can have very similar cellular components. Here, I'm just illustrating cytoskeleton, different types of cytoskeleton across different organisms. But you see that these homologs are just very similar cytoskeletons. They can, even though they are very similar in sequence and 3D structure, they can specialize in very different functions. So just studying how genes and proteins specialize, or they diverge, or they converge into similar sequences is not enough. You have to see in the cellular context what they're doing. So we're interested in connecting the molecular behavior, dynamics, and structure to cellular function, and then try to understand a little bit more how the cells evolved and interesting social behaviors, mainly. So as mentioned, I started my career working with bacteria, this oranging group over here, or you can say domain of life. And more I got interested as I got deeper into the molecular mechanisms. I got more and more convinced that only studying a few model organisms will be enough to really understand how cells select certain mechanisms, and then they regulate their assembly, and then they regulate the social behavior. So that's when I decided to move on to something that presumably was very similar. There is Archaea. These are microorganisms because they resemble, in the appearance, resemble bacteria so much. They probably, because of that, they were neglected for so many years. But now we understand that they are much closer to eukaryotes than to bacteria themselves. So there's an immense diversity there to be studied. And to start with, my lab decided to pick a very developed model organisms that are called Archaea. These are halophilic organisms that grow in salt ponds. This is one example in New Zealand, this pink salt lake right next to the ocean. And then you see this coloration because we have not only Archaea, but other microorganisms that secrete components that will bind to metals. and together with these beta carotenes, they give this coloration. But that's not why we decided to work with them. We decided to work with them because they have very straightforward genetics, similar to yeast. They grow relatively fast. There is a community that works and does like very good biochemistry and genetics. So as cell biologists, we got very interested with these organisms. And then another reason why I personally very interested is because climate change is compromising the salt lake diversity. And then it's gradually killing Halo Archaea. So it's kind of in a hurry to start isolating as many organisms as we can from these habitats before it's long gone. The prediction is in 10 or 20 years, they're going to be long gone. They're going to all die because of the higher and higher dehydration of these lakes. So as soon as we started studying these organisms, and with the help of many other labs that also study Halo Archaea, we started noticing that they are even more fascinating than we expected. We see, when we put it under the microscope, not only we see a variety of different shapes depending on the species that we are studying, we also starting to observe when we apply certain perturbations, they behave very differently than most of other organisms that have been studied in the past 100 years by cell biologists. So one obvious questions that we have is that how they create and maintain these shapes, how they regulate the cell division, and then why such like, very related, genetically speaking, organisms can assume such variety of shapes. And to advance on this understanding, my lab decided to not only focus on 1 or 2 species, but right now we are cultivating over 100 different Halo Archaea, distributing the whole taxa of Halo Archaea. And then you can see that we're starting to see a number of different morphologies that have never been reported before. It's very common for people to isolate new organisms, sequence their genome, report GC content, they report genetic traits, and then deposit and then report how to grow them. But there's not a single image, or images that they show are very hard to make any sense out of that. So we decided to push this work forward, and we have a student that is creating a Halo Archaeal atlas with all information that anyone that can access to any database can see: morphology, growth rates, size, anything that people want to know about Halo Archaea in terms of visual and growth physiology. We want to publish pretty soon. But to start studying these organisms and in such a variety and diversity, there are not only chemical considerations. This is the first thing that people think about. What kind of medium going to grow them? How long they're going to take to grow? Can I do genetics? This is a very immediate concern. But not many people think about the biophysical considerations of studying organisms under a microscope. So the first consideration is mechanobiology. Because this organism is growing such high concentration of salt, the way they found out and they evolved it to get around this high osmolality outside of them is to keep the osmolality inside the cytoplasm as high or equivalent to the outside, meaning they have low turgor pressure. So here's an example that is interesting. When this is a slow motion, 10 times is lower than the real time that we collect the data. You see when one cell gets certain momentum and hits a second cell, you see their cytoplasm retracting. And this is a very interesting observation, even though it's anecdotal at this point, that one, the cytoplasm seems to be very gel-like material. Second, there's not a lot of resistance because of this lack of difference between the outside and inside of cells. And another consequence of this is that when we use standard immobilization techniques to have cells stop it at a certain focal plane, so we can track molecules inside the cells, And then for prokaryotic cell biologists, agarose pads, conditioned media is the standard thing, and not only for prokaryotic cell biologists. And this is very effective for bacteria, but most bacteria do have turgor pressure, meaning they can fight back when they are gently compressed. But when you do this with Archaea, something very amusing and special and very annoying happens. They all deform, and different species will deform in different ways, but all of them lose their capability of propagating shapes and maintaining at least their biophysical properties. Our lab is very interested in read out these cells under different physical perturbations, and then treat them as materials rather than living things, and to understand a little bit more about like the material science behind all of that. However, I'm not going to talk about this today. What I'm going to talk more is about why we need to be careful when you image and make sure the results we get and the observations and quantitations and measurements that we collect from live cell imaging need to pass a certain criteria to make sure that they're physiological, not artifactual. The first question that we have is that why they are so squishy. And then the reason for that, together with them having like a low turgor pressure, is that they also don't have a rigid cell wall. Just like many animal cells, including mammalian cells, they are coated only by glycoproteins outside, but different than most of other organisms. These glycoproteins self-associate, no covalently, it's just protein-protein interaction to form symmetrical groups. And this is an example actually from a bacterial protein, not an archaeal one, but it represents really well what they do. This is the case, is a symmetrical group, a P6. So we have 6 molecules creating a 6-phase symmetry. And these little symmetrical units, they co-assemble in a monolayer, 2D crystalline lattice at the outer surface of cells. So we can think about this just as a 2D protein origami that coats the cells and provides some sort of a scaffold to reshape them. Even though this is still not very clear if these lattices are actually responsible for cell shape, we do have some indirect evidence. For example, different archaea that have different cell shapes, they will have different outer surface proteins that will self-associate in different symmetrical groups and organize the envelope in different ways. And when you purify these vesicles out of cells and coat them with vasolayer, you can see that they start producing this polygon and sharp angles again. So this is an indirect correlation, but it's not causation per se in the context of live cells. So, but an interesting feature about having this outer surface or S layer proteins coating the cells is that it makes them squishy inside, but they are quite rigid at the membrane. So if you photoablate cells and then break them, you're going to see that they can crack just like egg, get their cytoplasm leaked to the supernatant, to the environment, but they remain they can maintain their shape for longer period of times unless you apply any other perturbation to them. So this is also very different from most microorganisms that have some internal pressure, and then when there's a poke a hole or they break, they just pop because the flow that pushes outside of the cell is enough to tear the cell apart. So to get around all these complications to study these organisms, we decided to build, together with our collaborator, Jaroslav Icha from the Weizmann Institute, we decided to build a set of different micro and nano fabricated machines. I'm not going to get into the over dozen different machines that we created, especially for Archaea, but I can tell you that our machines, they have 3D tracks that can not only control where the cells go and different shapes go inside the chambers, but we can also control the flow of the inside this chamber to study the mechanobiology of them, creating more shear, creating compression. So we can actually control their environment and then screen for conditions that they're actually happy, which we advanced a lot at this point. And now we can, as you can see, we can see a lot of things that we didn't see before in real time at least. We can see different variety of shapes, and we can see actual propagation of these shapes. You can see some rods, you can see some polygons, and then we can use image analysis to study how cells propagate over many lineages. Again, I'm not going to talk a lot about this. I'm going to advance for a second issue that we work at the physiology course at Woods Hole this year. That is, when you want to study that many 100-ish different species at the same time, you have to have a better plan than just putting them in a microfluidic study one at a time. So one of the problems is temperature. So if you go to literature and then start studying and then and compiling the information of all of the isolate organisms, you're going to see that most of them report that they optimally grow around 37. And then there are some papers that report that they grow at like uncommon, temperatures. There's no way of actually knowing that, but talking to the dozens and dozens of labs that contribute isolating this directly. Some of them are not even around anymore. But our idea is that it's very unlikely that such diversity will select and convert to a very specific temperature. That it seems like not very that doesn't correlate with the environment so that they live in. So we decided to do bulk liquid growth curves of each one of them, screening temperature, and to our not so much of surprise, you see a nice variation of doubling time. I mean, is the time that it takes for a cell to double its mass, following by optical density in this liquid bulk growth curve. So we see that the diversity of growth regimes, depending on temperature, it's larger than reported. However, this doesn't mean that this is the optimal temperature by far. So we wanted to go even deeper into studying the single cell behavior of these organisms and screen for temperatures. So what we decided to do this in batch and then high throughput was to use VAHEAT substrates and bond our fluidic chambers. Here you can see one device only attached to VAHEAT. In the future, we expect to have dozens of devices connecting the same substrate for reasons that I'm going to mention pretty soon. But with this device, we are able to mix different cultures. They are labeled with organic dyes of different nature, and then we can feed these channels and these traps and image single cells while we ramp up and down temperature, like every couple of generation times. This way, we can have an immediate readout of growth rates really fast and cover a wide range of temperatures that VAHEAT also allows us because it has a great control of temperature. So we don't need to go to bulk and then do growth curves with dozens of plates inside a plate reader anymore. We can do this in few experiments and in batch, and then varying temperature in the same experience and see how cells respond immediately to temperature. So this is one example of one species that we track it. So each one of these points, the circles, represent one cell that we track it over time as we vary temperature. And this is basically a minimizing parameters to determine when cells don't grow faster anymore. So you can see that in some cases, we don't see a lot of variation from condition to condition, but then they get a huge jump right in the beginning of this exponential curve that we see that it's the ideal temperature of growth. Now, based on doubling time, we can come back, plot the single cell growth curves, determine these slopes, and then replot again the doubling time and compare with bulk. And so in purple, we're going to see the growth rates or doubling time of organisms that were now optimized by the right temperature. That's completely different than reported in literature. While the blue ones was the ones that we collected compared to the literature and we calculated in bulk. So it's clear that now we are able, no matter how divergent a certain organism is, we are able to optimize their growth in terms of growth rate, but also temperature. So we know exactly what we need to do when we choose a specific organism to study a specific behavior that we see under the microscope. So what is next now that we have such variety of different species? One thing that we want to study a lot is differentiation and specialization of function of different cells. Something really special that is reported for Archaea already is that it's not only that they have different shapes compared across different species. This, even if you get one species, you're going to see that they interconvert their shape between rods and what we call discs, plates, some people call polygons. You can pick your flavor there. And this variety of shapes is actually functional. It's important for their survival. You can see here rods swimming around the fluidics. This is a very slow rate. However, these discs or amorphous cells, they do produce pili, which structures that will attach to the glass and to the PDMS of the fluidics, and they will just colonize those places just like they want to create microcolonies, while you have these motile cells, they're rod ones that they probably prospecting new environments. And then we can, of course, report in the microfluidics the interconversion of one shape and another. So our next step that we're very excited to study with possibly with VAHEAT is to see if these interconversions, instead of following a chemical cue from the environment or nutrients like conditions, they're actually reading out temperature. Just like liquid droplets that they face transition to different shapes and even different behaviors when we ramp up or down temperatures. So this is just detergent. They're just lipid droplets with detergent. And then the only thing this group is doing, and let's just publish this paper, it is amazing, is to increase temperature really fast. This will create this will face transition to less organization of the lipids and creating this little tubes that look like they're propulsing the cells and pushing them ahead and making them motile. Of course, for cells, that's not how they become motile. They do have proteogenesis structures that push them ahead and make them swim. However, it's just impossible to ignore the similarities, even when you compare how fast the lipid droplets and change their shape, and also how fast archaeal cells change their shape. So we are convinced that there's a lot of biophysics regulating the interconversion of shapes. We are also interested in how the molecules inside the cells are also reshaping and scaffolding these shape transitions. So again, coming back to my initial slide, it's not only the physical cues, but it's how the cells evolved to select biological and biochemical mechanisms to respond to these physical cues. So for that, we are tracking cytoskeletal dynamics, And we are tracking also these outer surface proteins that I mentioned that create these lattices at the outer surface of cells. So we can see when these proteins are getting into the crystalline lattice, getting out, and what they're doing when the cells change shape. I think that I will stop from here. I want to thank our lab. It's great. Like, only in 2 years we are growing, and people have like such like different interests that I'm always proud to support. And I also want to thank like our funding agencies. We are very lucky that Archaea is on the spot and it's being highlighted a lot. And funding agencies are getting more and more excited about funding this kind of weird, squishy science. And also thank like Interherence again for allowing us to use VAHEAT for such long periods of time during the summer and start collecting this exciting data. This will actually support and promote a lot of very good science across many different organisms. And I would love to take some questions now.
Thank you, Alex, for an exciting talk and really great movies. We have a question in the chat, and then we have also one attendant raising the hand. So I will take the question from the chat first. We are exploring the temperature control systems for calcium imaging. Oh yeah, no, sorry. Let's move on to Francois Guillot. Now you should be able to.
Yeah, thank you. Can you hear me?
Yes.
Yeah, thank you, Alex, for this very great talk and fantastic images. I do not know in detail the setup of your growth experiments in the VAHEAT, but one common problem we encounter, I'm working on Archaea, but an hyperthermophilic Archaea rather. And one problem is that the growth rates, they really depend very much on the chemical composition of the medium. So I was just wondering whether this was an issue, and how did it change within the VAHEAT batch system?
Yeah, that's a very, very good question that indeed I didn't touch at all. So of course it's much more than chemistry, but it's also a lot about chemistry. So the thing is that with all this 100, we have like 115 right now growing species. It also makes the microfluidics helps us to rapidly screen different media and different composition and different concentrations of each components of the media. However, it's very surprising that right now we found that it's very, very easy to consolidate and start finding a common recipe for most of them, and to direct evolve, do like experimental evolution to adapt them for this universal media, even though we only need a few mutations in the chromosome for that. So we also have a student that is just doing experimental evolution for this.
If I am correct, these Archaea, they need oxygen, right? They need the dioxygen to grow.
Yeah, some species more than others.
Okay.
But for sure, they are very sensitive to oxygen, and because there's such a high concentration of salt, there's very little oxygen there. That's why also microfluidics is so great, because we are always replenishing them with oxygen much more than media, right? Media is very saturated with nutrients, so we don't see a lot of correlation of like late exponential growth and early. There is enough food there. The problem is oxygen.
Yeah, thank you very much.
Yeah, thank you for the question.
Okay, Senthil has a question.
Alex, very nice talk. Of course, I follow your work. You mentioned it's 1 or 2 mutations to adapt. Does that mean with temperature changes, is that the whole transition, or is that dependent on one protein? Because there's cytoskeleton and everything else as well, so they need to adapt as well. So I'm curious, from an evolutionary perspective or an adaptation perspective, what's the deal there?
Yeah, no, amazing question. And okay, first, like, no, great talk sent you. Like, you know, it was a very, very hard task to follow you after, like, you know, your data, amazing single tracking particle. But yeah, about there's not only one or two proteins. But depending on the feature that we are artificially evolving in our setups, we see different systems. And then it's mainly about biophysics. That's also reinforces us that so the mantra of our lab right now is that Archaea never evolved a rigid envelope because they have some advantage. And our hypothesis is that they are very, very capable mechanosensors. They sense everything that is physical around them. And this gives them this advantage. So every time we change something, media, temperature, right now we are evolving these cells to grow at low salt concentration. So these guys usually grow at 3.5 molar salt, the model organisms for Archaea. We evolved it for around 1 molar already, and we keep pushing. So we are learning the biophysics. How do you restructure and reorganize the cytoskeleton, the outer surface lattice, everything to adapt to this new lifestyle and behavior? And then, of course, we see correlation of different shapes. They are now unable to assemble anymore because of that. And we also see new shapes that were lost before doing evolution. And then because now we are adapting them to new context, they now can transition to these shapes again.
Right. Cool, thanks.
No, thank you.
So initially we promised to be finished at 5:10. So let's close the discussion here. I think we can continue the discussion per email with the speakers, and also if you're interested in VAHEAT, just get in touch with us or contact us through our website. And we are going to organize more online events like this. For example, another webinar in November focused on active matter colloids. So slightly different audience, but perhaps also interesting for some of you. And I hope to see you again online or offline on one of the microscopy related conferences next year. So have a nice rest of the day and goodbye.